National Cancer Control. They are interviewed by Amy Robach, co-anchor GMA3 What You Need to Know in 2020, and reporter for Good Morning America.
Around the world last year, 10 million people died of cancer and 20 million new cases were recorded. And yet, of all cancers that can be, yet half of all cancers can be prevented. That's the big thing we want to talk about right now, prevention. Here to discuss how we can unite to prevent cancer are Dr. Ron DePino and Princess Dina Myrid. Thank you so much for being with us, both of you. And Ron, I'm going to begin with you because we've talked a lot about how the COVID pandemic has exposed disparities that were already there, but certainly heightened them in the United States and around the world as well. So how can we use what we've learned during this pandemic to pivot into a healthcare system that does focus on prevention and detection?
Amy, thank you for asking the question. I think that there are three key lessons that we've learned. First and foremost, that science and technology, when applied, can profoundly impact the health of humanity. We've also learned that when we unite diverse stakeholders, those in policy, biotech, education, faith-based organizations, foundations and philanthropists, healthcare providers, we can dramatically accelerate change. And I think a great example of that are the COVID vaccines. And then thirdly, we've learned, painfully so, that an ounce of prevention is worth a pound of cure. And with respect to cancer, given the aging of the world population, we're going to have over 1 billion people over the age of 65 very soon from now. That's going to drive a dramatic increase in cancer. And the world cannot afford to remain in a reactive disease care system. We really have to shift more to a proactive health care system, which is really focused on the prevention aspects of diseases such as cancer. Now, if we're successful, we will profoundly impact the economic and societal fabric of the world, particularly for the underserved. This is a real challenge because there's an enormous growing gap between this transformative science, all of this knowledge, all of this potential, and the implementation of those solutions. So we really have to focus on the prevention aspects of disease.
When you talk about that gap between what we know and then actually implementing that knowledge, what is missing? What's the disconnect?
So I would say that this gap between knowing and doing really rests on the fact that we really have to collaborate. In fact, this conference is focused on bringing diverse stakeholders together, that uncommon table where you have different groups, each with unique capabilities, parts of the solution that when brought together can really move the needle on any particular problem. The second is that while disease typically manifests during adulthood, really the seeds of disease are planted during childhood. And so action needs to be taken during childhood in order to prevent adult diseases. And then lastly, we need a lot more information. We have to understand what the instigators are of disease, of cancer, at the neighborhood level, in communities, because different communities will have different problems. And so we need to help them understand what are the specific problems and what are the solutions needed to rectify those problems, remove those instigators. And that kind of granular data will enable us to asymmetrically deploy limiting resources. So I think those are all critical opportunities for us to really move the needle. And if we do so, again, we'll have a profound economic and societal impact.
And Princess Tina, I want to bring you in this because we have talked about the disparities in cancer fatalities between low and high income countries. And we're even talking about highly treatable, highly curable cancers. So talk about what you've seen firsthand and those gaps that need to be filled. It's a life or death situation.
Thank you, Amy. And I think this is spot on. It really is, with regards to cancer treatment and surviving cancer, it is a tale of two cities, basically. 70% of deaths do happen in low and middle income countries. As we speak right now, a child that may have, for example, leukemia, which we all know has about 90% chance for a cure. I'm a mother of a cancer survivor. In other countries, in Africa or other low middle income countries, that child has only about 10% chance of a cure, if that is if they do get a treatment at all. I visited countries that have a population in Africa, for example, of 40 million, and they have just acquired their first ever radiotherapy machine. Imagine, I even hugged that machine at one point because, they've just acquired it right now. And if you ask why did that happen, I think because way back when, I don't know who started that, they decided, the powers to be, that low and middle income countries are only busy with communicable diseases, infectious diseases, and that rich countries are the ones who are only facing off cancer, diabetes, heart disease, etc. And as a result, all of these countries did not, there was a big gap in the readiness to fight cancer and to actually build the infrastructure for cancer. And as we know, to build the infrastructure for cancer is expensive and it takes time. And that is why we are where we are. A lot of countries are still lagging in being able to have the human resources needed, the infrastructure, the machinery, you name it. And that's why, in a way, this conference is so important, because prevention is always the best way forward.
Ron, I wanted to talk about prevention and how late is too late. You talked about the importance of starting in childhood. I had cancer at 40, and that was about when I started to realize all the things that I had done wrong and really changed my diet, my nutrition, my exercise, all of that. But how do you tell people when it's okay to start? Is it too late to start at 40? Give us a sense of what the science is saying and showing.
Yeah. Very good question. So good news is it's never too late to start. And if you implement certain strategies, you could have a profound impact on your health and well-being, the incidence of disease, and your life expectancy. Just to give you some details, just 15 minutes of exercise per day can increase your life expectancy by 3 years and reduce the incidence of cancer, diabetes, and heart disease by about 15%. Now, if you do 30 minutes, you get even more benefit. Managing stress, chronic unrelenting stress, and lack of sleep can have a very significant impact. Engaging in preventive services, so, mammography, colonoscopy, skin exams, these are enormous opportunities to really catch cancer at its earliest, most curable stages. So those are all really important opportunities for us that can really make a big difference.
Right. It's something that gives people the power to say, I can do something individually without a large cost to myself to make a difference in whether or not I live or die or how long I live. Princess Dina, I once had someone tell me there's a cure for cancer. It's called cash. I'm curious, with your experience around the world, what is the biggest problem or the biggest hurdle to either short-term or long-term solutions for cancer? Is it money? Is it political willpower? What is it?
Of course, you do need resources. It is a big issue because cancer management is expensive. We all know that. However, it's not just about money. I speak from my own experience here in Jordan when we, as a team, tried to fix our hospital that was literally tantamount to a morgue. And then we worked to fix it to become what it is today, the fantastic King Hussein Cancer Center. Our problem was not money. Imagine, we had the infrastructure, we had the machines, we had the human resources. It was purely management and governance that was the big block. And throughout my visits, when I was president of the Union for International Cancer Control, I saw the exact same thing in low middle income countries, whereby, for example, maybe a country had, imagine, a new radiotherapy machine bought, but the room to house it was not the right size. I mean, management, governance, that kind of thing. Political will is key as well. If you don't have political will to actually fight cancer and to provide the services for your own people as a country, it's really tough to make things happen. Also, the donor communities, the way they behave, that also needs to change. they come in and they want to do, for example, early detection of breast cancer. They wanted to do that with us in Jordan. And we said no, because it wasn't at the right time. We were focused to fix treatment per se, in, firsthand, because we felt, why would we tell a woman about her breast cancer to make her aware if she did have breast cancer when we hadn't even fixed treatment yet? And so we knew what we want to do and when we should do it. Things like that. Also lobbying by the food industry, by tobacco industry, and so on and so forth, who are completely, they are a huge element in not helping countries being able to fight cancer. And again, when we talk about prevention, tobacco comes number one. In my own country, we've had huge lobbying by tobacco industry to prevent us from doing all the things that we should do to reduce consumption. So, but then again, prevention, I always tell countries, prevention is key. Every $1 invested in WHO evidence-based best buys, exactly the kind of things Ron talked about, physical exercise, reduce alcohol, tobacco, etc., will yield the return of $7 on NCD prevention, including cancer. So actually, it is a win-win if there is political will to actually, utilize the knowledge and the tools that we have now.
Yeah, that makes a lot of sense. And go ahead, Ron.
I think that just to underscore the really important statement that was just made, we cannot afford not to invest in prevention. Just to put it in very sharp relief, and with HPV, a virus that causes cervical and throat cancer around the world, in the United States alone, there are 34,000 cases per year, and it costs us about $8 billion to treat those diseases. For us to vaccinate all 4.4 million 11-year-olds in the United States each year would cost about $1 billion. And that might actually go down if we go from 2 doses to 1 dose in a trial that's currently underway. So it's not that we shouldn't be investing in this. We cannot afford not to invest. So resources are important, but also leveraging diverse stakeholders. Knowledge is cancer's greatest vulnerability. So if we use our education systems to teach our children about healthy living and prevention, that could impact families. Policymakers, with one stroke of the pen, could have a profound and lasting impact on health outcomes for their citizens and so on. So I think the theme of this whole session here is about bringing diverse stakeholders together, each with their responsibilities, their unique capabilities, into a collective impact that really moves the needle on the prevention opportunity that we have.
And Princessina, I know you've done a lot of work when it comes to making, or at least ensuring, that the HPV vaccine is something that is universally accessible. Talk about your work there, and talk about, the communities that are hardest hit by this type of cancer that is preventable.
Absolutely. I actually now sit on the WHO task force for the elimination of cervical cancer. I mean, where do you get is disease right now that you can say you can eliminate? Cervical cancer is one. You can prevent with a vaccine, you can early detect, and you can treat. So this is a great opportunity to help save the lives of 570,000 women, that's the last number from 2018, who have cervical cancer. And again, unfortunately, 70% of which are in low middle income countries. So we do have an opportunity to do something and save these women. And, we can do it. Again, we've seen, as Dr. Ron said, vaccines being produced at maximum speed. This is something we've never dreamt, that they can produce these vaccines at this speed. They can do this. We need to watch about how to disseminate these vaccines in an equitable manner so that all can receive the vaccines, not just high-income countries, as unfortunately that we've seen right now, countries, nationalism, racism, you name it. We have to learn from that if we're going to make this happen, and to make sure that every girl and everyone has her rightful chance to have the vaccine. But there is a big opportunity because we can remove one disease off the map, and this is great.
Yeah, that is very incredible to hear. And I'm going to end this with you all each giving me, for about 30 seconds each, the 3 tips you would offer adults, what they could do right now to greatly reduce their risk of getting cancer or even dying from cancer. Ron, I'll start with you.
Well, I would say engage in prevention strategies, so screening and that sort of thing. Obviously, if you're smoking, stop smoking. Secondly, would be to be more active and eat healthy, and the third thing would be to manage stress proactively, and it can be done.
Go ahead, Princess Dina.
I'm not going to repeat what Dr. Ron said because I agree with all of that, but I also say, really believe that your health is literally in your hands, that you can prevent getting cancer and other NCDs by 30 to 40%. That's on you, on the individual.
Princess Dina, Ron, thank you both for your incredibly wise words and your impassioned speeches. We all got a big takeaway from them. Thank you.
Thank you, Amy, and thank you, Dr. Ron.
Pleasure and honor.
This discussion around precision-guided care features Dr. Christian Tomasetti, Dr. Andre Gua, and Dr. Sigal Kadosh, and is moderated by Dr. Otis Brawley.
Hello, I'm Otis Brawley of Johns Hopkins University. It's a pleasure to be here today and talk about cancer with Dr. Christian Tomasetti of Johns Hopkins University, Dr. Sigal Kadosh of the Dana-Farber Cancer Institute in Harvard University, and Dr. Andre Goy, who is from Hackensack Medical Center. Let me start out first with you, Christian. You've done a lot in terms of the molecular biology and genetics of cancer. What have you discovered in terms of what causes cancer?
Background mutation rate is essentially the fact that every time a cell divides, will accumulate some mutations on the DNA. They estimate 3 to 6. And so, but this background mutation rate was not really thought to have a particular role in cancer. What we discovered is that, in fact, the more cells, the more cell divisions, and therefore more of these replicative mutations, which are random, accumulate in an organ, the more the organ will have a lifetime higher cancer risk. And also, when you look at cancers and you do sequencing of cancer tissue, what we discovered is that you could essentially attribute about 2/3 of all the mutations that you find to this background mutation rate, which is what I call bad luck. So essentially, we discovered that this normal endogenous process that's happening, continuously in all of our cells plays a major role in cancer.
Now, Dr. Goy, you're an expert, one of the world's experts in precision medicine. How has precision medicine evolved, and how has our greater understanding of the genetics of cancer, and cancer as a genetic disease, helped with the evolution of precision medicine?
Sure. The 3 traditional ways and modalities to treat cancer have been radiation, surgery, and chemotherapy for over a century. And the progress in understanding cancer cell biology, as well as boosted by the Human Genome Project in the early 2000s, as well as the decrypting of the role of the immune system in cancer, has really profoundly transformed cancer for a number of reasons by largely increasing the number of therapies, from targeted therapy, immunotherapy that now gives option to patients that would have been sent to hospice before, and also shed light on the extreme diversity, molecular diversity of cancer at the biological level. And it'll help identify markers that can predict a response to a treatment, and that we refer to as biomarkers. So this molecular diversity can now be appreciated through routine molecular diagnostic that we have mostly researched before, next-gen sequencing in particular, that can be done at the point of care, particularly from liquid biopsies, because we can find in the blood the DNA, RNA abnormalities in particular that reflect the cancer. This has made the managing of cancer much more complex and increased the number of options, but also the obvious need to tailor the therapy based on the genetic makeup. And that's the foundation for precision medicine.
Thank you. Now, Dr. Kadosh, Sigal, you've done these marvelous 3D models that help us to understand what's going on inside of a cell. Can you describe these genetic models a little bit and tell us what they're used for?
Sure, absolutely. And just as my colleagues, Dr. Guo and Dr. Tomasetti, just elegantly described, the sequencing of the human genome and subsequently cancer genomes has immeasurably advanced our understanding of the molecular, specifically genetic underpinnings of human disease. And that's certainly the case for cancer. And just a couple of years ago, we saw the fruits of a very large scale effort called the Cancer Genome Atlas, where they sequenced multiple different tumor types and identified thousands of new putative missense mutations, new drivers. And many of these were expected culprits that have been longstanding targets for drug discovery efforts for many, many years. But some of them were very unexpected. And one of the most surprising and unexpected findings to come from these large-scale genetic studies in cancer has been the idea that chromatin regulatory processes, chromatin biology-related processes, are frequently perturbed in cancer. And our laboratory studies these large, as you mentioned, Otis, large macromolecular assemblies, large, big machines that sit on chromatin and essentially regulate the topology or the architecture of the genome. And our lab is interested in studying those because the genes that encode the various components of those large regulators are mutated in over 20% of human cancers. That's a significant burden of cancers. And in addition, several rare but genomically well characterized cancer types, such as synovial sarcoma. This is a disease that we've spent a lot of attention on, malignant rhabdoid tumor, and a number of other diseases that have been increasingly identified through sequencing. These are driven uniformly, 100% of cases or near 100% of cases, by perturbations in this large machine. And so we took it upon us to really understand, how are these molecular machines pieced together in the cell? How are they assembled and loaded onto chromatin, onto DNA? And what do they look like? What's their 3D structure? So we've had a lot of investment in time in understanding that, as a way to then understand the underpinning mechanisms that can give rise to new strategies for therapeutic intervention.
Now, Christian, what are the implications for all of this in terms of cancer?
Well, so one major implication is that I think up to very recent times, the idea was the cancer was just due to environmental exposures and inherited factors. What this research has shown is that, in fact, there are important endogenous processes, and so there is also civil war going on at the same time. So what does this mean? What this means is that, one of the major implications is that, of course, primary prevention is fundamental. It's the best way to do prevention. But there may be a part of cancer that we will not be able to eliminate through primary prevention. And so what do you do about that? And this research seemed to say that it may be a relevant fraction of it. And so the idea is that if you cannot do primary prevention, if you cannot avoid the disease from occurring, you can avoid the disease from killing the patient. Okay, and so that's called secondary prevention, essentially through I think the major way to do it is through early detection. 5-year survival rates, for example, of cancers like ovarian cancer, okay? We go from 90% when the cancer is being detected locally to 30% survival rate when the cancer is being detected, in an advanced stage. We need to put a lot more efforts into secondary prevention and early detection in particular. And I think these are very exciting time for that, because there is a lot of research and even a lot of companies that are investing, billions of dollars in this. So this is not just the opinion of some scientist in his little office. This seems to be really what, even the market is thinking is going to be a major next step.
I can't help but think it's almost 50 years ago. It was December of 1971. When Richard Nixon signed the National Cancer Act. That's when Senator Ted Kennedy said Richard Nixon declared war on cancer, and we got that war on cancer analogy. The three of you really are instrumental in that war, in that you're helping to understand what cancer is. And as we understand what cancer is, we can understand both how to reduce risk of getting cancer, intercept cancer when it is early and someone can truly benefit, and then treat cancer when it's late. Now, Andre, you're using you're using the cancer immune system to treat cancer. Can you just tell us a little bit more about that and get into this wonderful, what is CAR T cells?
CAR T cell stands for chimeric antigenic receptor, and this is a receptor that is introduced to lymphocytes, the cells of the own cells of the patient, to force them to recognize and attack cancer cells. So this is the first live therapy. The cells are briefly taken out of the patient, manipulated in the lab, expanded, and are reinfused about 2 to 3 weeks later. What is remarkable, it has the activities in relapse refractory B-cell lymphoma leukemia is 80-90%. And we had a recent update, and we have been involved in this for years at John Theurer Cancer Center. Recent update of the first pivotal trial in aggressive lymphoma, where 40% of the patients are alive 4 years later, in a situation where the survival or median survival in that setting would be 3 to 6 months. So we have now 5 different approvals for lymphomas and leukemias. Myeloma will likely come this year. There's a huge exponential increase of interest in CAR T cell and T cell engaging therapy in general, right? With antibodies, bispecific, but focusing on CAR T cell now, we have learned on how to mitigate the toxicity better by being preemptive, and we're going to put a switch gene over time, trying to make better CAR T cell that have a better T cell fitness early on in the disease, and then also to make dual CAR T cell or tandem CAR T cell that can prevent resistance. What is remarkable in this, the durability of this response in patients who had no option. And we have some patients that were part of the first trial we did with the NIH in the mid-early 2000 that are 8 years out. And the first patient that was called Emily that received CAR T cell with an ALL is almost 9 years out. This is unprecedented. So this is a game changer. We are very obviously excited to be part of this. And it is a therapy that took 30 years in the making to be the first live therapy ever approved.
You know, it's quite gratifying. Andre, you and I are about the same age. We came into medical oncology as clinicians when our tools were really very limited. And we've had the experience of being able to literally see where our profession has moved to the point that we can do some very positive things. You want to talk about that? Doesn't it really feel good to be an oncologist right now?
Yeah. I actually moved into oncology because I was interested in a specialty that would be closer to the lab and that would move fast and that would be still intensive in a way. So I focused on blood cancers. And happened that my mother was my first patient and was a junior fellow. And then I didn't want to treat her. I forced them to treat her. That was my first patient. She did really well against the expression of ovarian cancer. That's certainly a life-changing event. But to your point, I went into this because I thought it would be a dynamic specialty. I didn't expect it would go so fast. It's just unprecedented. And I think the future is really, really bright. I just want to add one thing on precision medicine, that there was so much resonance with the precision medicine or personalized medicine that there was a lot of frustration because the expectation was that we're going to do a gene signature, you're going to be cured. Well, I think this is really prime time now, precision medicine. And we heard from Dr. Tomasetti that in a pre-cancer early detection, this is going to be very important. 100% of cancer stage 0 are curable. And 60% of cancer deaths have no screening test. So this is really important to understand. The other area, beside obviously giving the right treatment time of cancer, is the post-cancer care, where precision medicine will help us evaluate MRD and immune status and prevent recurrence. So this is going to apply across the whole phase of cancer care, and I believe is the solution to our problem in cancer.
Mm-hmm. Now, Christian and Sigal, you represent the scientists who actually helped us understand what cancer is, and you helped us to understand how to manipulate it. Okay, and for you made the cancer problem into an engineering issue, and Andre and I are the engineers who apply the science that you've created. Look into the crystal ball, look into the future. You've given us so much over the last 40 to 50 years in terms of understanding cancer. What does the future hold? Sigal, you do things like, I hear these wonderful words, CRISPR. Tell us a little bit about what's going on in the laboratory and what you're able to do.
Sure. So I think we've seen an explosion in the technologies that can be applied to defining the underpinning molecular basis for cancer, this certainly holds a great deal of promise for precision medicine. In addition to the sequencing that I described earlier, that has really revealed for the first time what are the molecular determinants of cancer, that's given us as scientists a real roadmap. It's the reason why a basic scientist like myself has chosen to study this particular entity. It's because we're inspired by the results of these large-scale genomic studies, which have implicated such frequent mutation frequencies in this category, or this class, if you will, of genes. This is unexpected. By understanding that, we know now that, parts of breast cancer, or subsets of lung cancer, subsets of ovarian cancer are driven uniformly by problems in this particular entity. Before, it was just considered breast cancer, lung cancer, ovarian cancer, and I think increasingly we're seeing additional subtypes that are going to be extremely useful for the translation of this biology into the clinic. We're already seeing a number of targeted therapeutic approaches in the very area that I'm describing, really understanding how can you molecularly disrupt these master regulators that control large numbers of genes, so gene programs. And I think the understanding that a large number of cancers are driven by these master regulatory factors starts to begin to explain why we saw some major challenges in what I call the sort of gene expression era of precision medicine, or the drug discovery, where really, there was identified to be, high expression or low expression of a certain gene, and that was going to become a target. But really, it was one of many, and we just didn't have a way of understanding that yet, because we didn't know that there was a larger player upstream of all of those controlling the expression of large, of many programs. And so I think by hitting these master regulatory entities within the cell, particularly these nuclear entities, holds a lot of promise going forward. And in parallel to that, you asked about CRISPR. These technologies, the ability to alter the genome and understand even the consequences in the laboratory of altering genes at specific residues, altering genes and knocking them out. These are large-scale screening efforts that have been performed now by many, many groups. This has told us for the first time what are the dependencies, what are the synthetic lethalities of tumors that were previously unappreciated. Where are there some very surprising, oftentimes, molecular events that are keeping that tumor proliferating and keeping it intact? How could we then alter those? How could we then target those? So this has really brought forward a number of new therapeutic targets that would have, before this era, not been able to be identified.
So on my side, I think one of the most exciting developments is the multi-cancer screening test. So let me just describe a little bit what that is. Every time a cell dies, some of the DNA may be shed in the bloodstream of the person. And that, of course, is not just true for normal healthy cells that die. It's also true for cancer cells, luckily for us. And so what happens is we call this cell-free DNA. It's basically fragments of DNA floating in our bloodstream. And when a patient has cancer, in several cases, a small fraction of that cell-free DNA, it's fragments from cancer cells that have just died. And so in very recent years, and I think this is one of, in early detection, maybe the most exciting news is that, there have been developed some technologies, and I'm working on them with at Hopkins with a group here for detecting this small fraction of fragments that come from cancer. It's a very difficult game, as you can imagine, because often these are just 1 out of every 1,000 fragments, or even every 10,000 fragments. But I think we are there. We and other groups have published recently results where you can detect about up to, I think, medians across studies say something like 70% of cancers can be detected by these methodologies. And so what I see in the near future, and when I talk about near future, I don't mean, sometimes we talk about technologies that are promising and we think about 20 years from now, 30 years from now. I actually think this may be, within the 5-year range from now. At least this is what I'm hearing. We may have like a simple doctor visit, our early doctor visit, where we take a blood test, a regular blood test, a few $100s, and we can detect at an early stage a cancer. I think that's like really an extremely exciting, future thing coming.
Yeah. Now, machine learning, computers have been incredibly important as we start learning this. And also, I'd like for you, Christian and Sigal, genes versus genomics. Can you just tell us a little bit about the difference between genetics and genomics, and how has machine learning and computers helped us with all of this?
Sure. So one can imagine the vast amounts of sequencing data that have poured in from all regions of the world over the past decade plus now has been a major computational challenge, I think, for the world at large. And certainly computing has played a major role in being able to handle that data, be able to process that data, and increasingly make it easier for people without the extensive knowledge of handling that data to be able to call up gene mutations, for groups without computational expertise to be able to understand their experiments that are using genetic sequencing. Genomics applies to all of the approaches that at some level leverage DNA and leverage DNA sequencing. So this can include what is known as epigenetics as well, processes that happen on DNA, so the topology of DNA, proteins, understanding the interactions of proteins that are governing the architecture of our genomes. And of course, that's important for dictating the timely expression of genes, whether genes are turned on or turned off at the right times. And of course, cancer and other diseases is often a failure of that process to go correctly. So we use a number of genomics approaches where we are trying to actually profile where proteins sit along our 2 meters of DNA. If you take the DNA out of each one of our trillions of cells, it would stretch 2 meters. Of course, it's less than the size of a pinhead if we absent to the naked eye, and that's because it is compressed several thousandfold to fit in the size inside the size of a nucleus that we can't even see. And so these processes have to be there to allow for this compaction. And so we need to understand what are all the proteins that around which DNA is wrapped and spooled to be able to bring that compaction together and to be able to open DNA for genes to turn on. So those are genomics approaches that can be used. There are a number of other approaches considered genomics, again, using CRISPR. This is affecting genes. So it is genetics, but it can also be used as genomics to target. So I think we're seeing this unique pairing for the first time where genetics and genomics are often together. And you actually can see that reflected in the idea that many departments throughout our institutions, throughout the country and the world have now merged those two because they're really playing off one another very often.
Maybe a comment on machine learning very quickly. I think that every time we have had a major scientific revolution, it's been because of our ability to see better something, in more detail. I'm thinking about in Renaissance and pathology, when finally in Padua and other places, they started opening the human body and really looking inside. I think this is what is happening thanks to bioinformatics and machine learning. We are looking at something that's extremely highly dimensional. And a person on its own would have a very hard time figuring out any of it. And so these methodologies, these approaches are helping us. The blood cancer test that was mentioned, it's one perfect example. It's thanks to these methodologies that we are able to see these very small signals.
Yeah. In the last few minutes, Andre, you and I train the next generation. Are you optimistic?
I am fully optimistic. I think that we are the crossroad in cancer care, in medicine at large, between the demographics, the increase of cancer care that will be 45% more in the next 20 years, and the sustainability and the cost of healthcare. Digitalization of the society, we heard about data, machine learning, etc. And the only revolution in precision medicine, immunotherapy that I mentioned. I think we are at a crossroad and we need to really redesign and look at what we're doing. There's already plenty of opportunity, and I think the future is much more diagnostic than therapeutics. We have already plenty of treatment options. We don't do great analytics yet in healthcare, evaluating the individual outcome, pick among all the options that we have. And I think this is where the future has to be, is really share data, and be able to really drill down and understand for each patient what is the best option. And the biggest question in cancer care now is what is the best sequence of care? There are so many options. As we speak now for lung cancer, there's more than 80 options to treat advanced stage lung cancer. So how do you develop an outcome really that helps rationalize the care? And I think that's a solution to improve the outcome of the patient and reduce the cost for the population.
Thank you so much. It's been wonderful to be with you today. Thank you, Dr. Tomasetti, Dr. Kadosh, Dr. Goy.
Thank you very much.
Dr. Brawley.
Thank you, Otis.
That was great.
Heroes are not born, they're made. Occasionally, they're thrust into that role suddenly. Often they grow into it slowly, sometimes taking many years to show their inner ethos that was there all along. That was the case with Helena Thurer. Born in the Washington Heights section of New York City, Helena attended George Washington High School, which is where her formal education ended. Her parents passed away when she was young, so Helena had to support herself, taking on a variety of jobs from Wall Street to bookkeeping at small companies. As she says, if I wasn't learning at a job, I moved on. She was managing the office for a small truck manufacturer when John Thurer bought the company and made Helena an offer she couldn't refuse to come work for him. It took a few years, but eventually John made another offer Helena couldn't refuse. He proposed. Together, they grew the company John's grandfather had founded as builder of horse-drawn wagons in 1888 into the second largest manufacturer of truck trailers and shipping containers in the U.S. But with John's health failing, the couple sold the company and retired to the Florida Keys, all the while acquiring extensive real estate holdings there and back in New Jersey. When John passed in 1994, Helena, the self-taught accountant and real estate magnate, continued to astutely manage her holdings through economic downturns and recessions, then slowly selling them off until today the only real estate she owns is her home. Helena knew she had been blessed with a loving, if too short, marriage and the wherewithal to share her good fortune. So Helena embarked on a voyage of philanthropy, with a gift to the medical center that was founded the same year as her late husband's truck company. Her first gift was dedicated to the memory of her late husband. It helped establish the John Thurer Cancer Center at Hackensack University Medical Center. Realizing the benefit her gift has had for the thousands of cancer patients treated there, Helena then decided to go all in on philanthropy with an additional $25 million gift to help build a modern pavilion that would help bring together exceptional patient care and medical innovation. This powerful combination would extend her impact on healthcare and research beyond cancer, pushing medicine beyond traditional thinking and providing patients with amazing possibilities. That building and its legacy will bear her name at Hackensack Meridian Medical Center. But Helena's impact goes still further. Her belief in giving back and paying forward will encourage others to share their good fortune. As she likes to say, I don't need all the money I have to live. I should share it. And I hope it inspires those with, quote, a little extra cash here and there, that it's okay to give it away, for a good cause, end quote. And so we award Helena Thurer with the Pontifical Hero Award for Inspiration as a recognition of her generous philanthropy and unwavering dedication to improving the lives of patients and winning our collective fight against cancer. Medical science is making extraordinary strides against a wide range of diseases, from rare genetic diseases to common but no less deadly maladies such as heart disease and cancer. Perhaps no therapies have had a greater impact thus far than those broadly defined as immunotherapy. It has now enabled treatment and even cures for cancers not amenable to the classic surgery, radiation, and chemotherapy triad. But cancer immunotherapy is a very broad field, encompassing a variety of checkpoint inhibitor drugs from Genentech, Roche, Bristol-Myers Squibb, Merck, and others that essentially take the brakes off a patient's own immune system, allowing it to attack the cancer. Another exciting immunotherapy is known as CAR-T therapy from Novartis, Kite, and Juno, among others. This entails genetically modifying a patient's T cells to recognize tumor cell markers, and they've been curative against certain leukemias and lymphomas. A number of companies are also developing CAR-T approaches to solid tumors. However, possibly the area of immunology that has most captured the public's imagination is stem cells. Their potential, still not well realized, is immense for everything from cancer therapy to growing replacement organs in the lab, regenerating and repairing worn, aging, or damaged tissues, and even cosmetic applications. But procuring enough cells to meet the potential demand, particularly if they must be matched to the tissue type of the recipient, will be challenging. That is where the unique characteristics of placental cells come in. Those extraordinarily plentiful cells are not only generic, meaning they needn't be tissue typed, they may also be an abundant source of natural killer or NK cells. NK cells can destroy tumor cells, as well as virally infected cells, as in COVID-19 disease. This is a special three-part session on novel approaches and platforms for treating disease. Part 1, Revolutionizing Cell Therapy, features Dr. Robert Hariri, founder and CEO of Cellularity, and Cellularity Director, Dr. Andrew Pecora. So let's start out with, there's a lot of work being done in cell therapy, and that's a pretty big tent, right? You've got stem cells, you've got NK cells, CAR-T cells, all kinds of different cells. Let's talk about the particular area that Cellularity is working in, which is placental cells. That, I think, is a little bit of a surprise to many people. What is it about placental cells that are unique and that makes it you guys where you really want to focus your energies?
Well, Max, the three of us have really been actively involved in this field for the last 2 decades. And what we are very grateful is that there's been progress made in cellular medicine in the area of immunotherapy for cancer, and that's driving the entire field forward. What we recognized many years ago, Andrew and I in parallel, was that for cellular medicine to become a meaningful part of healthcare, someone was going to have to figure out a way to productize, to pharmaceuticalize living cells. The ideal source material for the platform from which you create these medicines, these cellular medicines, just turns out to be the leftovers of a full-term healthy pregnancy. At the dawn of cellular stem cell biology and cellular medicine, the concern was you might have to use embryonic or fetal materials, which are very problematic for many, many reasons. But consider that the placenta is essentially nature's stem cell factory, and 130 million times a year in the world, this organ is disposed of. It's just the perfect raw material. So simply enough, the placenta is where we can get stem, progenitor, and even mature cells like immune cells that can be turned into medicines.
So Andrew, most disease processes involve cells either mutating or somehow losing their normal function, or what we would normally consider their normal function. How do placental cells fit into this? Do they correct the aberrant cells, or do they just replace them? How do they fit into that framework?
So, Max, that's a great question. When you think about it from a holistic perspective, if you're using a cell therapy to treat a disease, there are two basic things that you want to accomplish. In diseases where the immune system is abnormal, meaning it's overactive like lupus or rheumatoid arthritis, you want to tamper down the immune system. In other diseases like cancer, where, as you suggested, you have a mutation, and that confers to those cells an unrestricted growth ability that can just grow like crazy, what we call cancer, you want to kill those cells. What's unique about the placenta, it has the obvious ability to regulate the immune system because it exists in the mom for 9 months and keeps the fetus healthy. So as inherent within its properties, its native properties, the ability to have a normal immune system, not an overreactive immune system. And what we have learned and what we have productized is the ability to take some of those cells and turn them into effective killer cells, NK cells, that can kill cancer cells, and that they're not antigen restricted, meaning that you don't have to have a target to go after. They can go after anything that's cancer, and they can kill virally infected cells. And the beauty of NK cells, particularly placental NK cells, is they can go after any virus. So whether or not it's COVID, a mutant of COVID, or a bad flu or anything else. And as it relates to T cells, those are antigen restricted. But when you have an antigen that's unique to the cancer, like CD19 for ALL, as an example, you can target CD19 and not hurt the patient and get rid of all the cancer cells.
So is it fair to say then that these cells that we're talking about not only are, as you said, not antigen restricted, meaning they're kind of generic. We don't have to tissue match them like you would in a stem cell transplant, right? And cells seem to know what they need to do. It sounds like, you put them in the right situation and they kind of figure out, well, I need to regulate the immune system over here, or I need to kill these bad guys over there.
Take this one, Bob.
Yeah. The way we look at it is nature's a lot smarter than we are. The cells from the placenta are designed, as Andrew pointed out, to be a universal donor cell and tissue that allows it to exist in an unmatched recipient and take on the functionality of cells that might be defective or deficient in that recipient. Now, interestingly enough, the fact that placental cells are, for lack of a better word, they're very young and naive. They lend themselves very well to all the manipulations to enhance certain functions, like as Andrew pointed out, going after a specific target. They also, by virtue of that naivete, they are less likely to induce any of the immunologic reactions that cause complications, things like graft versus host disease. And we have built over the last 2 decades great experience in the tools necessary to manipulate and alter the functions of those cells, but the sky's the limit. Andrew and I have been talking for over 20 years about the future of cellular medicine being that if you have a platform of cells that's reliable, scalable, high quality, and has acceptable economics, you can literally apply all of the breakthroughs in genetic modification and engineering, et cetera, in order to drive them towards a therapeutic use that's meaningful in a disease, whether it be an immune disease or cancer.
And Max, one of the important things that is going to become a mainstay in this conversation about cell therapies is a fundamental concept in biology called stemness. The more stemness you have, the more you can do. When you think about it, it's just obvious. The fertilized egg, how many replications does that need to go to with fidelity to result in a fetus, then a baby, then an adult that lives for 90 years, as compared to cells taken from a 50 or 60-year-old? There's log order greater stemness from placental-derived cells than any other cell source that you can have. And that's
more capacity to become different tissues.
Well, not just that, not just that, that's true, but even more than that, the ability to grow rapidly and maintain their integrity without genetic mutations, without getting tired, without running out of gas. if you take a cell from a placenta, that's going to turn into an adult man or woman that lives 90 years. All of that biologic potential is housed in these cells, and we have figured out how to harness that power, that stemness, and turn it into therapeutics for the greater good.
And you know, Max, this is becoming so increasingly relevant, not just to things like immunotherapy, but even to the future applications in other more mainstream diseases. As Andrew pointed out, the ability for a cell to go through that number of cell cycles and that number of cell divisions and still maintain its synthetic and differentiation integrity is incredibly important for the future of cellular medicine.
Another important aspect, I think, here, we hear a lot about, particularly in cancer therapy, CAR T cells, right? And they're manipulated, genetically manipulated and engineered, but they are bespoke cells. I've always wanted to use that word in a sentence. Bespoke, one-off cells, right? They're one of a kind. With the placental cells, we don't need to take and do one treatment that is unique for each individual patient.
Yeah, that's correct, Max. And this was the brilliance of Bob Harari some 25 years ago. He was prescient in his view that instead of having to have a single patient be their own source of material, which is this so-called bespoke model, it's complicated, it's expensive, it stresses out the patient because they don't know that their cells are healthy enough. These are heavily treated cancer patients. Their cells may never make it back to them, even in time. Whereas Bob's view was, let's create an allogeneic source that could be good for anybody and could be used whenever they're needed.
And even better than that, good for anyone, meaning that you don't have to mix, you don't have to match, but it's a one size fits all product. But also, just from what Andrew described, this ability to essentially repopulate an entire human being until they're 90 means that starting material in the manufacturing environment can produce so many doses that one placenta donor can serve thousands, potentially hundreds of thousands of recipients. That scalability is vitally important to this becoming part of a meaningful therapeutic regime. So, you know, the cells from the placenta, readily available raw material resource. Remember, we collect placentas at 40 weeks gestational age, plus or minus 2 weeks. The biological consistency of that source, compare it to adult autologous donors who have to provide their own cells at a time they've been diagnosed with disease. No two patients are the same. They're different ages, they have different biological histories, different epigenetic modifications to their cells. So imagine you have highly variable entry material into a manufacturing process in autologous CAR T. What comes out is going to be highly variable. In comparison, placental cells go in with a great deal of biological consistency, and what comes out at the other end is also consistent.
And you brought up manufacturing. Manufacturing cells and turning them into a therapeutic is not the same as making aspirin or acetaminophen or any number of other medications. How do you handle that? How do you make sure that you've got the same, that you in fact have the same product coming out of the other end of the manufacturing process?
You know, it's a great question. When we first embarked upon this, our dream was that we were going to figure out a way to expand the number of cells from a specific donor into quantities sufficient not only to treat them, but to treat many other patients. And we needed to create a system for that expansion that was extremely reliable, wasn't dependent, for example, on enormous outside reagents and factors, and could ultimately be reusable and consistent between donors. And that's what we've done here at Cellularity. Our manufacturing environment uses a highly choreographed process from which the raw placenta, the organ itself, is used to isolate cells that are the starting material. Those cells go into, in essence, a cultivation environment. Think of it like a bioreactor. And the cells expand, they duplicate and duplicate, and we do it so that they don't necessarily get driven to differentiate, meaning that they maintain that stem cell phenotype characteristic. Then we isolate those cells and then can drive them down different lineages if we want, for example, immune cells, or they can be used in their pluripotent stem cell form because those cells even have their own biologic characteristics that are useful medically. But the manufacturing, and Andrew is an expert in this as well. He's created many companies in the space. The manufacturing investment companies had to make to be able to make reliable products has been enormous. It's taken decades.
And one of the important points, Max, getting back to your point before, is that because of the stemness property of these cells, they grow like weeds. 1 million cells, within a month, you have hundreds and hundreds of billions of cells. So one placenta can result in the products for hundreds, if not thousands of patients. So the cost of goods sold, the economy of scale, to turn this into a true pharmaceutical is a reality because of the inherent property of the cells.
And remember, if you look at alternative sources, adult-derived cells that have to be driven to expand and proliferate, or in the case of induced pluripotent cells, they have to be tricked into behaving like stem cells. All those efforts are unnecessary when you're starting with material from the placenta that already does these things and doesn't have to be engineered and manipulated and has a great deal of biological consistency. So that stemness and the fact that this raw material resource is so abundant makes our original discovery of this organ as a source of these cells, we think, quite important.
And Andrew, we're just about out of time, but before, right at the beginning, you mentioned NK cells and some of the other uses that you might put to. Give me a little shopping list, because this sounds so exciting. What's the shopping list of ways that you foresee being able to use these cells?
So genetically modified T cells to go after specific targets when that's appropriate. NK cells to go after generic targets when that's appropriate, either cancer or virally infected cells, and mesenchymal-derived pluripotent cells to reestablish proper immunity across the broad array of human illnesses. And then the final frontier is regeneration of damaged tissue or tissue that's aging.
And one last word, Bob, how far off is the future?
You know, Max, first of all, we're very, very happy that we're now in an environment, Andrew and I now are in an environment where we're increasing the pace and the tempo of development and innovation and invention and so on. I actually think that in the next 3 years, we will see a massive increase in the number of immunotherapy products available for cancer and immune disease. That is going to catalyze more activity in the use of these cells in degenerative diseases. And then all of us, it's relevant for the three of us, we are looking to the future where this same source will provide cells that will actually preserve our performance, and preservation of human performance is the next frontier for cellular medicine. And that means that individuals can receive essentially augmentative therapy where these cells support the failing or deficient populations that are no longer doing their job, fixing our cartilage, making our hair grow, et cetera. And that, I think, that's the horizon that I care a lot about.
Max, I think he called us old.
He's the one with the gray beard. I don't know. Bob, Andrew Pecora, thank you. It truly does sound exciting. I look forward to the future very soon. Thank you.
Thanks, Max.
Great to see you, Max.
While cell therapies have generated much excitement due to their diverse and robust potential in regenerative medicine, many biotech companies have taken what could be called a reductive approach. Rather than entire cells, they're working with molecules that are a different type of immunotherapy. This class of drugs has been around for some time in the form of monoclonal antibodies for therapeutics, diagnostics, vaccines, and more from pharma giants like Amgen, Genentech, GlaxoSmithKline, Bristol-Myers Squibb, and dozens of others. Regeneron and Eli Lilly have made headlines with their monoclonal antibodies for COVID-19. More recently, in an effort to improve on the effectiveness of monoclonal antibodies, companies have explored what are grouped together as small molecules, although some are still quite large. One such class that is truly a novel approach to treating disease is called bispecific antibodies. These compounds allow binding to two or more different receptors, potentially bringing together disparate targets and effectors, either cellular or molecular. Roche, Merck, and AstraZeneca are exploring their use beyond oncology to include a wide range of applications, from autoimmune disorders to arthritis, psoriasis, and asthma. But other truly small molecules are also seeing a flood of research interest in drug development. They've been called a revolution in pharmacologic chemistry because of their many unique mechanisms of action and diverse applications. A unique small molecule class that has been dubbed the next blockbuster therapy goes after what were once thought to be undruggable targets, disease-causing proteins. Targeted protein degradation utilizes small molecules to destabilize such pathogenic proteins by tagging them for dismantling by normal intracellular processes. While virtually all major pharma companies have a stake in this class, smaller biotechs such as C4, Arvinas, and Chimera are also actively pursuing its use. Few companies have as large a presence in the small molecule therapeutic and diagnostic space as Sorrento Therapeutics. Henry G., Sorrento's president and CEO, joins us now to discuss their technology. What's your approach, a comprehensive solution, if you will, for COVID-19?
So, hey, thanks, Max. And our solution to it is detect early, treat timely. Treat timely means a different stage of the COVID-19 disease. You treat with different measures or treatment solutions. So, for example, we had the COVID-19 rapid antigen detection system called the COVID stick. And we can detect whether you have infection or not in 2 to 5 minutes. And we can excluding you are negative by waiting for up to 15 minutes. Now, with this detection, you can detect the patient from symptom 1 to 7 days. The moment you have symptom, you say, okay, you're infected. Now, what's the solution? Next step. Next step is we have a neutralizing antibody, because it's neutralizing antibody is the most effective way to treat the COVID-19 infection, because the neutralizing antibody binds to the virus and to remove them and blocking them from replicating into the cells and replicating and spreading. So we have a two form of way to administer, actually 3 ways. One way is to have IV infusion, or we call IV push, because we only require 2 to 5 minutes instead of 1 hour, 5 hours infusion, you have to be hospitalized. So it's very convenient outpatient. The other form of neutralization administration is using intranasal drops or nasal drops. So that means you can drop the things into your nose, and they block the virus into the cells on the epithelial layer in the nose, and blocking them replicating into lung tissues. And that also blocks the spreading. Mm-hmm. Another way to do that also is having a genetic material encoded plasmid DNA and inject into your muscle cells, like your muscle, have the muscle to express the neutralizing antibody in your body. We actually have government DARPA grants to support this program, and this is a very rapid measure. And for example, right now the variant is, very concerning, and like a UK variant, South Africa, and the Brazilian strain. And we have a deal with Mount Sinai, got an exclusive license for all their antibody dealing with all of the mutants. So we have a cocktail right now of 2 antibodies we can put in together in the 3 different forms, IV form and intranasal form, or into a genetic encoding material into your arm in the muscle. So express all of these neutralizing antibody and blocks almost all of the variants as of known today.
So Henry, let me ask you the diagnostic part of it. How is that administered? What do you use for the diagnostic?
This is very simple. You do a nose swab, very simple. It's not deep nose swab, but rather shallow. And you put it into a lysis buffer and pull into a detecting cassette. And the things positive, negative, it shows up right away.
So, So you mentioned also then that this cocktail that you're testing that can be administered a variety of different ways. How is it that this can cover all of the different variants then?
Yes.
Since we keep hearing that the variants are very specific for the vaccines, how is it that this covers the different variants?
Because we test it. We have all of the variants, we have their protein, we have their virus, and we test it. If one of the antibody or 2 of the antibody works on it, and we right now detect a couple of antibody we can put together as a cocktail of 2 that blocks all of the virus of concern, every one of them, including original Washington strain, the UK strain, the Brazilian strain, the South African strain as of today. And then eventually virus gonna be run out of tricks. So if we discover enough antibody covers all of them, and then you can, feel safe because we have a cocktail of antibody blocks them all. Now,
and as new variants pop up, do you manufacture these new antibodies, or how do you then decide what to put into the cocktail?
Now we believe right now we have enough antibody covers all of the mutants of this moment. And if anything emergent, we just need to discover more antibody and just to make sure the antibody does not escape in our antibody cocktail. All right. So now with this said, the next step, if you get to already infected, develop the syndrome in the mild to moderate, and this neutralizing antibody really works and on the outpatient setting so that you have the diagnostic, is infected, you have your doctor do telemedicine, give you a prescription, you potentially go to a CVS Rite Aid. From the pharmacy, you pick up the COVID drops and the intranasal drops, or you can schedule into outpatient setting, do an IV push, and that gives you the treatment. Now, what if you already develop a little bit more severe, let's say moderate to severe, in the ICU, what happens? So we have solution 2. One is oral pill, dealing with the cytokine storms, and that is called abivertinib, it's a BTK inhibitor. And we already have about 140 patients enrolled in both US and Brazil. And we're gonna get into big time in Mexico. So this drug, if that works, gonna be potentially discharged from the hospital very quickly. And then if this gets really severe, like if people gets into ICU and need oxygen and intubated on the ventilator, we have mesenchymal stem cells, which is derived from adipose cells, right now in human testing. And this right now, every one of the patient using this infusion, which it requires 3 infusion, every one of the patient treated discharged from the ICU. And some of them on the deathbed. After first infusion, feel much better and requires less oxygen. Second shot, they want to go home. Third shot, they immediately released from the hospital. Wow. We're talking about discharge from ICU and the discharge from hospital on the same day.
Wow. So
very impressive.
This sounds obviously very exciting. And it sounds like you have the capability to develop some of this for other viruses or even other diseases, infectious diseases, and going on to cancer.
Yes.
How else will you use this platform?
Yes, so we have a human antibody library approach. The approach does not require the recovered patient samples. For example, all the neutralizing antibody discovered from infected patient recovered with that neutralizing B cells in their body. However, this is in vitro in the test tubes. It's a test tubes with billions of antibody in it. So we can just screening any target. Let's say you have a nipple virus just happens right now, and we can have the sequence and putting the antigen in it. Immediately, you're gonna get, within 2 weeks, you're gonna have antibody. Now, if we do plasmid way to do it and encoded the plasmid into your muscle cells for muscle to express, we can have this response, rapid response within a month. That's where the Department of Defense, DARPA, give us the grant to aggressively, getting this one tested, validated as a rapid measurements response for any emergent pathogen. Of course, this can be used for antibody therapeutic in the oncology space. For example, we can use the plasmid DNA to express the PD-1 antibody, which is about Keytruda is selling for 15 to 20 billion, Optivo, another 12, 14 billion. You're talking about 60 billion product we can express in your muscle, let yourself express it. So that would disrupt the whole business, the industry. So we have enzyme replacement for Gaussian. Now that could be interesting too.
So how you have so many different possibilities here within the company, how do you decide what to focus on? You know, the saying that a jack of all trades is a master of none. How do you decide where to really put your energies as a company?
Yes, so we're all staged. For example, diagnostic side, we already submit the EUA. If the FDA gives us a green lights, we start in general revenue. And now we already have this neutralizing antibody in the phase 2 study, and abivertinib in phase 2, and mesenchymal stem cell in phase 2. And these are rapid clinical testing. Could be getting to a point to potentially EUA approval, all of them in this year. So that gives you a potential revenue generation. And everybody say you have a lot. We have a big pharma's portfolio. Now we have a very strong resources in the public equity as well as cash level. And now we can do quite a few programs at the same time. Now, hopefully one day we're not only jack of all trades, master of none, we could be a master of a few.
So you can chew gum and walk at the same time.
Exactly. That's
Henry G, thank you so much for your time. It sounds very exciting, and we look forward to hearing more about it.
Thank you very much, Max.
From innovative drug development strategies, science has also delved into the very core of our cells in a search for novel approaches to treating disease. It has taken 2 decades, but the sequencing of the human genome and the technologies it has spawned are now beginning to bear remarkable fruit, first for rare diseases and now for common afflictions from heart disease to cancer. Probably the best known genetic intervention, CRISPR, is examined in another conference session, but there are a number of other nucleotide paths to disease treatment that may be as productive as gene editing. Most are permutations on that now renowned molecule, RNA. Companies like Biogen, Novartis, and Alnylam are turning the tables in a way on RNA by impersonating this vital nucleic acid, but in a way that hinders its normal function. Broadly termed antisense oligonucleotides, they bind to specific sites on functional RNA, blocking its ability to make a protein or work in other ways. But no application for RNA has had a greater impact on human disease in the 21st century than messenger RNA, mRNA for short. It is an incredibly powerful and flexible platform that is being harnessed not just for COVID-19 vaccines, but for a number of other vaccine targets as well. Then there's therapeutic applications to make replacement proteins in rare genetic diseases and manufacturing drugs against cancer. One of the earliest companies to see and explore the transformative potential of mRNA is Moderna Therapeutics. Here to discuss his company's varied uses of what he calls the software of life is CEO Stéphane Bancel.
what is different about an mRNA vaccine that you've developed versus some of the more traditional, I guess, virus-based vaccines?
Yes. So, Max, it's good to see you, and thank you for the invitation. So in the case of an mRNA vaccine, we basically, in our factory, make a piece of messenger RNA so that it can be injected in your body, and your own cells are going to read that message. And from the instruction on the message, it's going to make a protein, in the case of this vaccine, the spike protein, so that your immune system, when it will see that protein, will make a lot of antibodies, so that if later on you get a natural infection of a virus, you will have already antibodies to bind to it, so that you don't get sick and the virus doesn't replicate in your body.
So essentially, you're using our own bodies as as a manufacturing plant.
Oh, exactly. And the way to think about it is mRNA is software, mRNA is code, which again, it's a new concept to a lot of people, but the way life works in plants, in fishes, in humans, in animals, on the entire living kingdom on the planet, the way life works is all the information is stored in DNA. We think about DNA as the hard drive of people's instruction set. And then mRNA is a molecule in between that makes a copy of only the instruction for one protein. All your proteins are in your DNA, but mRNA, only one protein to make the protein into your body. So it's really a piece of software.
Terrific, terrific. So using this new technology, you've been able to create a vaccine that the numbers look like 95% effective in a remarkably short period of time, perhaps less than a tenth of what it normally takes. What was it? Was there any one or what things allowed you to move that quickly?
So a few things. The first thing is mRNA, because it's software, it's a platform. So the way we make vaccine against flu or against Zika or against the SARS-CoV-2 is exactly the same technology. We only change the instruction coming from sequencing, getting the genomics information of the specific virus, and then we drop it in the same cassette, literally on the computer. It took us 2 days to go from the sequence posted online by the Chinese to have finished the design of a vaccine. Usually it takes years in the labs, it took us 2 days. So that's the first piece. The technology is really quite remarkable to be able to copy and paste and just do it again and do it again and do it again. The second piece is this was not our first vaccine in the clinic. This was actually our 10th vaccine in the clinic. So we've gained experience, we've improved the technology, we improved the manufacturing process over the last 5 years that we have been doing this in the clinic, but the company is 10 years old now. The third piece is the FDA. The team at the FDA has been remarkable. The collaboration with the FDA has been fantastic. We had a team dedicated for Moderna. And unlike in regular times when a company asks a question to the FDA, they have up to 30 days to decide a date to schedule a meeting in the future. We had answers the same day, on weekends, in the evening. Sometimes we had 2 to 3 days per week. That same process will take maybe 3 months in normal times. Wow. And so very quick dialogue with the agency to allow us to make decisions that were really critical to get to the next step. And I would say the last thing is the US government. I have to give a lot of credit to the US government last year, and Operation Warp Speed was actually on the vaccine front. I can only speak about the vaccine piece. It was really remarkable because by giving us a grant of $1 billion, like they did to all the big companies that are working or have vaccine authorized, that allowed us to take a lot of business risk. Obviously, it was out of the question to take clinical risk on participants of the studies. That was just a no-go. That never crossed our mind, that never crossed the FDA mind. But what we did with having access to that money is to take a lot of business risk. We've done a lot of things in parallel that usually you will do one after the other, because if something fails, then you will not waste the money of the next step. But when you are in a pandemic and you get a grant from the government, the reason they gave us those grants to all those companies is to say, we want you to save every day you can, because every day matters, because we are losing Americans every day.
So Stéphane, I know that Moderna, as you said, this vaccine was not your first entry into the vaccine world, but you've also been working using the platform for other therapeutics. What lessons have you learned now in this past year developing this that you'll be able to apply to some of these other areas, and what are they?
Yeah, so I think there's two types of lessons that we learn, Max. One is around the portfolio of a vaccine. On the vaccine front, we are so very much more willing to take calculated business risk now on our vaccine portfolio because it's exactly the same technology as the vaccine that has been authorized. So if you think about Moderna, in the last 10 years that the company was around, we believed we could, by investing in science, one day get a product authorized by the regulator. But we didn't know. We believe. Today we know that we can get an mRNA vaccine authorized by the regulator. And in business, between believing and knowing is a huge difference of your willingness to take a risk because you know this can be approved. So you will see a lot of acceleration of our vaccines and also a lot of very innovative vaccine we're going to bring to market that's going to benefit a lot of people. On the therapeutic side, which is making treatment, not vaccination, treatment for when people are sick, I think what has really helped us tremendously is the manufacturing scale-up. You know, this year we're on track to make up to 1 billion doses. That's a lot of material, and it's an incredible manufacturing achievement. In 2019, we only made less than 100,000 doses for the entire year. 100,000 doses for the entire year. I can tell you the face of my leadership team in manufacturing when in February of 2020, I told them, guys, next year we need to make 1 billion doses. They look at me and say, what are you talking about? It's impossible. We're like, it's 10,000 times more product we have 12 months to get ready. I say, yes, and people are dying, so let's get to work. And they find a way. They were very creative, extremely collaborative. They really did an amazing job. And that's for me a really tour de force. I mean, if you think about it, go to any amazing company, go to Apple or go to Tesla and ask them next year, you need to make 10,000 times more product. It's not like 5%. I think it's a really remarkable achievement that's going to benefit all of our treatments.
So you now know how to get the mRNA into a nanoparticle that will survive being injected. And for things like therapeutics, for treatments, all you really need to know is what's the protein that will either fix a problem or treat the problem, and you're off to the races?
Yes, it's that powerful, Max. You 100% get it. I mean, a drug that I'm very excited about, that I love to talk about, because one of my grandfather died of a heart attack, is a drug we are developing with AstraZeneca, which is now in phase 2 in clinical studies, being injected in people's heart after a heart attack. So as many people know, if you have a heart attack and you survive it, you will have very severe heart failure because your heart muscle will have been damaged by lack of oxygen during the infarct. And because of that, most people after a heart attack that they survive live between their bed and their sofa and their TV because they cannot even go for a walk because the heart has been so damaged. So what we are trying to leverage with AZ is regenerative medicine in your own body of a protein you have. We all have in our DNA, again, the hard drive of life, we all have the instruction of a protein called VEGF, VEGF. That protein, what does it do? It grows brand new blood vessel. We use it in our body every time we cut ourselves because our body has to heal by making new blood vessels to bring blood and oxygen and nutrients into the cells. And so the very innovative idea here is to inject mRNA once, 10 to 12 injections done by a cardiologist in the area of your heart that was damaged by lack of oxygen during the infarct. It has to be done within a few days.
And new blood vessels grow into the damaged area.
So we've shown that in mice, we've shown that in pigs, published in Nature, showing beautiful data and improvement of the ejection fraction capability of your heart. The phase 1 done by AstraZeneca also has been published. It's beautiful. The safety profile is pristine. And we see expression of VEGF, and we see increase in blood flow. I was on in people's arm as a way to de-risk, if you want, not going first in people's heart after a heart attack.
Right.
Very risky endeavor, of course.
Stefan, before we run out of time here, because there's so much here to talk about, the hot topic now is when it comes to COVID, are these variants that keep popping up. And of course they will, as you said, as we've discussed before, they will continue to pop up. What can you do about it?
Yes, so the variants, the good news is some like the UK variant, no impact on the vaccine. South Africa and Brazil, a little impact, but still very high level of antibody, way above what is required. So we think the current vaccine is very protective against those, but we don't know what the future is made of. And especially what will happen with waning immunity over time. And so we've announced a few weeks ago that we have developed already and made and shipped to Dr. Fauci at the NIH in only 30 days, a new vaccine that to be used as a boost, a single dose boost, with the exact full copy of the virus first identified in South Africa, that mutant. And we're going to get this one authorized if the data is good, which we anticipate, because again, it's like software, sometime this summer. And we are committed to chase any variant of concern.
So that as these new variants pop up, which they likely will, you'll be able to generate a vaccine against them.
Correct. And we will also be able to combine things. If down the road you have a UK 3.0 and a Brazil 2.0 that is an issue, we can have in the same vaccine, in a single dose boost for you, the RSA 1.0, the UK 3.0 and the Brazil 2.0. We will adapt because we can mix mRNA. We've already done it in the clinic.
Stefan, that is encouraging, incredibly encouraging news. Thank you for your time. Keep up the good work. This is really terrific. Thank you.
Thank you so much.
We have just heard from Stefan Bancel about the transformational mRNA platform pioneered by Moderna. But this level of science doesn't happen without a visionary individual who can lead and motivate a talented team to deliver on the promise of the science. That leader is Stefan Bancel. Born and educated in France, Stefan believed that so much innovation was happening in the U.S. that he applied to several graduate schools in America. That led to a master's degree in biochemical engineering from the University of Minnesota. The choice, he says, was partly due to less expensive tuition at a state school. Later came an MBA from Harvard Business School, various executive roles at Eli Lilly, and eventually becoming the CEO of the French pharma giant bioMérieux. Then in 2011, Stefan got a call from a friend, Noubar Afeyan, CEO of venture capital Flagship Ventures. Afeyan had been trying to lure Stefan to run one of Flagship's startups, but this time he presented Stefan with such exciting science that he was willing to take a huge career risk, something that, if it worked, would change the world. Thus, Bancel became employee number 2 of Moderna. The company's mRNA platform was initially directed toward therapies, a way to induce a human to make a protein in the body to cure a disease. The first targets were rare genetic diseases of children, and then cancer treatments. But he soon realized that they also had a powerful technology for vaccines, including Zika, pandemic flu, and cytomegalovirus.
The mystery virus started here in the city of Wuhan.
2 patients from the same family in England have tested positive for coronavirus.
Then in January of 2020, Stefan started seeing data that convinced him this novel coronavirus would soon become a global infectious scourge. He pivoted the company to go all in on a COVID vaccine, risking massive amounts of capital, even with the US government's infusion of cash from Operation Warp Speed. The speed with which Moderna was able to produce a viable candidate vaccine and enter human clinical trials was orders of magnitude faster than any previous vaccine. Despite the audacious gamble on an unproven vaccine, Stefan Bancel is quick to credit a dedicated team that he says sacrificed birthdays, family events, and more to work literally around the clock 7 days a week to bring the Moderna vaccine to fruition. He is especially proud of their commitment because they knew their Herculean efforts would be so impactful on human life across the globe. we show our true gratitude by awarding Stéphane Bancel and his team with the Pontifical Hero Award for Inspiration for leadership and steadfast dedication to protecting all of humanity. The groundbreaking gene editing technology called CRISPR has raced from concept to human trials with breathtaking speed. It was first described by researchers Jennifer Doudna and Emmanuel Charpentier in 2012, and it was so revolutionary that they were awarded the Nobel Prize in Chemistry just 7 years later. CRISPR is a kind of cellular toolkit borrowed from the defense systems of bacteria. It holds the power to add, remove, or alter and repair genetic material. This toolkit lets researchers do gene editing faster, cheaper, more accurately, and with higher efficiency than existing methods. In 2019, Victoria Gray became the first patient to undergo CRISPR treatment for sickle cell anemia. She is thriving and free of the pain and blood transfusions that had ruled her life. CRISPR is even being deployed to offset some of the effects of climate change. What used to require a decade or more to breed a plant to make it more resistant to a given threat, CRISPR can do in a year or two. For example, researchers have altered the genes of corn to grow at shorter heights to better withstand storms. And they have genetically engineered rice to resist 95 different strains of bacteria that cause blight. In humans, more than 50 clinical trials of CRISPR are underway. A dozen of them have advanced to phase 2. They target leukemia, lymphoma, HIV, myeloma, sickle cell anemia, bladder cancer, and more. In China, CRISPR is being tested against lung cancer. And in a first-of-its-kind case last year, a patient with congenital blindness received an injection directly into the eye, using a virus to deliver CRISPR to the retina in the hopes that the enzyme will repair a defective gene and restore vision. Plus, new CRISPR-based diagnostics are being tested for pneumonia, tuberculosis, whooping cough, and COVID-19. CRISPR is an acronym that stands for Clustered Regularly Interspaced Short Palindromic Repeats. It's actually a technique borrowed from an ancient defense system bacteria used to defend against viruses. The CRISPR complex consists of a strand of RNA bound to an enzyme called Cas9, which essentially unzips the double helix structure of the invading virus's DNA and chops it up. The power of CRISPR lies in the specificity that comes from lab-synthesized RNA, called the guide RNA, that will determine exactly where the gene editing will take place. Once the CRISPR toolkit reaches the cell's nucleus, it bumps along the genome, attaching every time it comes across a short sequence known as a PAM. At those PAM entry points, the CRISPR grabs onto the cell's DNA so the Cas9 enzyme can unzip it just a little bit. Then the guide DNA slips inside this unzipped double helix to look for the target sequence of DNA it seeks to repair, rewrite, or delete. If the guide RNA fails to find the right sequence, CRISPR moves on to the next PAM entry point, and the next, until it finds a precise match. Then the Cas9 enzyme cuts the DNA right where researchers were targeting. Then the cell's natural DNA repair mechanisms swing into action. Cells usually repair a break in their DNA by gluing the loose ends back together. It's a sloppy process that often results in mistakes, but mistakes can be beneficial if they disable or inactivate a disease-causing gene. CRISPR also lets researchers insert a custom-made DNA sequence into the newly split gene. It serves as a template as the cell repairs and reforms its double helix. That's essentially a new gene that could code for a vital, missing, or defective protein. Cancer is another CRISPR target, using gene editing to engineer CAR-T and natural killer cells that boost our immune system to make it better at destroying malignancies. Other diseases that may become CRISPR targets may include cystic fibrosis, muscular dystrophy, Huntington's disease, atherosclerosis, and progeria. The possibilities are endless. This is why CRISPR technology is so powerful and so very promising. But like any powerful technology, CRISPR's potential can be put to fraught uses. We must ensure that humanity's ethics are safeguarded as we explore this potent science. The conversation bridging science and faith has begun. CRISPR is one of the most exciting new gene therapy tools of modern medicine. In this conversation, Dr. Samarth Kulkarni, CEO of CRISPR Therapeutics, speaks with Edward Tentoff, Managing Director of Piper Sandler.
Hello, my name is Edward Tentoff, and I'm a Senior Biotechnology Analyst at Piper Sandler. I'm honored to participate in the 5th International Vatican Conference. CRISPR Therapeutics is the leading developer of CRISPR-Cas9 gene editing medicines. Based on the discovery of Emmanuel Charpentier and Jennifer Doudna, who were both awarded the Nobel Prize in Chemistry last year, CRISPR has demonstrated the curative potential of CTX001 in sickle cell disease and beta thalassemia. Additionally, CRISPR is advancing a pipeline of 3 gene-edited CAR T therapies to treat cancer and has a rich preclinical pipeline behind these therapies. With us from CRISPR today is my good friend Sam Kulkarni, CEO of CRISPR Therapeutics. Sam, thank you so much for being with us today.
Thank you for having me.
So pretty incredible that the Nobel Prize was awarded just last year for this technology. Perhaps you can start off by telling us how CRISPR-Cas9 works.
The discovery and development of CRISPR-Cas9 is a remarkable story. It turns out that bacteria have developed over millions of years a pair of molecular scissors that they use to inactivate viruses and protect themselves against viral attacks. In fact, I brought a little model of what a CRISPR-Cas9 system looks like, and it's probably what you see here is a Cas9 protein, which is the molecular scissors wrapped around DNA, and it finds a way to cut the DNA in a specific place using a barcode. So this is a system that Dr. Emmanuel Charpentier and Dr. Jennifer Doudna discovered and developed in the 2010-2011 timeframe, and they adapted this to be able to edit genes or DNA in any location across all genomes, whether it's animal genomes, plant genomes, or human genomes. And this was a remarkable discovery and development that spurred the potential for CRISPR-Cas9 to make a huge impact in medicine. And CRISPR Therapeutics, our company, was formed on the basis of access to this technology.
That's very helpful, Sam. So please describe CTX001 and how you are using CRISPR to cure sickle cell disease and beta thalassemia.
Sickle cell disease and beta thalassemia are terrible diseases. These are genetic diseases. In other words, they're diseases caused by mutations in the genome that are passed on from parents that have been carrying it. In sickle cell disease, you have one mutation in the code of life that causes hemoglobin molecules to polymerize and cause the red blood cells to form a sickle shape that don't flow well within the arteries and veins and cause pain, cause strokes, and ultimately early death. Beta thalassemia is also an equally deadly disease that's seen across all parts of the Mediterranean and also in various parts of Asia and Africa. CTX001 is a medicine that utilizes CRISPR-Cas9 DNA editing to potentially cure sickle cell disease and beta thalassemia. And the way we do this is we take the patient's bone marrow cells, send it to our manufacturing facility, where we make an edit using CRISPR-Cas9 in a particular place in the genome, and that elevates fetal hemoglobin, which is an alternative form of hemoglobin that makes up for the deficiency or defectiveness of beta globin. And when you put these edited cells back into the patients, they should be symptom-free and cured within a month or two of being transplanted with these modified cells. In fact, last year, one of the patients we treated, Victoria Gray, was a subject of an NPR story and article. This is a woman who is 34 years old at the time of being treated, with 3 children, living a life of pain and a life where you have the constant threat that you may have hospitalizations. And she had 7 hospitalizations coming per year coming into the trial. And after treatment with CTX001, after a couple of months, she's been symptom-free since, and now we've followed that patient close to 2 years, and that effect has been durable, and she's living a normal life, going from a life of pain and comorbidities.
Just incredible, Sam, really amazing. And it's so interesting that you're using the patient's own cells to actually cure them. Now, you recently reported what I think were just remarkable data at the American Society of Hematology meeting. Maybe you can walk us through some of this data and show us really about how long these responses are occurring.
We were very pleased to present data last year at this meeting, where we showed data for 10 patients that we treated with CTX001. And remarkably, and three of the patients were sickle cell, 7 patients were suffering from severe thalassemia. And remarkably, all 10 of 10 patients were symptom-free within 2 to 3 months after being treated with CTX001. And what we saw was that their fetal hemoglobin levels went up as predicted to overcome this deficiency or defectiveness of beta globin. And clinically, these patients went from requiring, in the thalassemia case, 1 to 2 transfusions per month to not requiring transfusions at all. And in the case of sickle cell disease, these patients who were being hospitalized and having to go to the emergency department every 2-3 months, and they went from that situation to not having to go to the hospital at all. While these data are early, we see tremendous potential from an approach like this to potentially provide cures to these patients suffering from these terrible diseases.
Absolutely incredible. And the thing that I think is really important too is that you are fundamentally changing these patients' lives, and there's all of the long-term effects that are going to be relevant with addressing the underlying disease. What do you see as the potential regulatory path here, and what are your goals to really bring CTX001 to patients around the globe?
Yeah, one thing I will say is, it takes a village to bring a new technology platform like this to patients globally. And it requires the support of regulators, the health system, and it takes a lot of effort from the company side, but also the physicians treating for these patients and patients themselves. I would say that we're operating with a great sense of urgency to get these CTX001 to approval, and ultimately to get to patients across the globe. With therapies like this, where the effect size is so large, it's almost binary. You get, you potentially going to get a cure after one treatment. You don't need to do trials with hundreds of patients. I mean, these are smaller trials, and we're already at the stage where we're in a pivotal trial, which potentially could be registrational. And so if you get to approval with this trial, we would move very quickly to expand our manufacturing, scale up our manufacturing, not just in the US and Western Europe, but across the globe, and bring this to patients in a safe and effective manner.
That's great. Now, Sam, what are some other diseases that could be treated with CRISPR technology?
One of the things we're very excited about is the area of cancer. It's been nearly 60 years since we declared this modern war on cancer, and we try to throw all the medicines that we had at our disposal to try and make to cure cancer or make a dent in patients suffering from cancer. But it still remains a death sentence for most forms of cancer. What's exciting about the CRISPR-based approach is you're retraining your immune cells to fight the cancer. So what we do is we take immune cells from a healthy 25, 30-year-old and send them to our manufacturing facility. And there we use CRISPR-Cas9 to modify the DNA, and we retrain these immune cells to go recognize a particular form of cancer. And once we inject these cells into the patient's body, they go and find the cancer, where the cancer is lodged, and kill the cancer cells. And we saw some very exciting early data that we presented last year with the first 11 patients suffering from a blood cancer, where we saw that these allogeneic retrained immune cells called CAR Ts can go kill these cancers. And so we're now pushing forward aggressively across a number of different cancer types where we have these re-engineered and retrained immune cells. And I think that's going to have a dramatic impact on cancer because we're now talking about an intelligent drug. You know, it's no longer some toxic chemical where you're losing your hair and you have a lot of other toxic side effects. You have a smart drug that's a cell that's been programmed to just kill the cancer and do nothing else in the human body. And so we're quite excited about moving forward our trials here. And we've now dosed a number of patients, and we hope to update further this year and next.
Yeah, we're looking forward to more data from those programs. What about the field of regenerative medicine? How can CRISPR technology be applied to treat some diseases in that area?
This is another exciting area where regenerative medicine means that you can literally regenerate organs that are defective. So if you have a defective kidney or defective liver, today you have to go into this long queue to find an organ that can be transplanted into your body. And most patients actually don't find a donor or an organ that can be transplanted. What we're doing here is synthetically creating these organs based on pluripotent stem cells and engineering them to form the organ of interest. In our case, our first foray is in diabetes, where we take these pluripotent stem cells and direct their fate to become pancreatic islet cells. Our pancreas are typically a banana-shaped organ where most of the cells are doing a different function, but a small percent of the cells are islet cells that produce insulin in response to glucose in our bodies. And when those cells become defective for various reasons, they're no longer producing insulin. So patients become insulin dependent, and these are severe diabetic patients that have to inject themselves every other day or every week. What we're doing here is taking these stem cells, making artificial pancreas, and putting them in a small device the size of a matchstick, and implanting that device into your flanks, under your skin. It's a very simple procedure. And then those cells live and grow inside your body and serve as your new pancreas, and that can secrete insulin in response to glucose. And that should be a durable solution for a number of years. While it's still in preclinical stage, we're tremendously excited about this approach, and we're going to start working in the liver. We're going to ultimately start working in the area of heart disease as well. And this notion of regenerating organs is going to be very powerful in medicine 10, 15 years down the line.
Yeah, you can really see how CRISPR technology represents the future of medicine. So Sam, with such powerful technology, how do you ultimately see CRISPR changing medicine?
It's going to change medicine in a couple of ways. In fact, I'll use the words transform medicine. One, I think there's thousands of rare diseases where people are born with a single mutation or a couple of mutations that happen, and it's unfortunate for those patients. And they're born with these rare diseases that are oftentimes deadly. In fact, one of the diseases we're working on, for instance, is called glycogen storage disease, 1A, where babies that are born with this disease have to be fed cornstarch every 3 to 4 hours throughout their life. And if they miss that, they could die. And it's very difficult for the parents. It's very difficult for the babies and the kids suffering from these diseases. And there are thousands of these diseases. And using CRISPR, you have the potential now to stamp out these diseases and give all these people or patients a normal life. So that's one way to transform medicine. The second is medicine's going to feel very different, whether it's heart disease or cancers or even neurodegenerative diseases. You know, I think today we're used to a world where we're taking chronic pills and medicines every day or every week that only treat the symptoms and not the underlying disease sometimes. And here we're talking about a new world of cures. You know, and you're going to come in for a one-time procedure akin to the medical device world today, where let's say you have faulty hips and you come in for a hip replacement. You come in here for a single procedure where either you replace the bone marrow or insert a new organ into your body, or engineer or inject these smart immune cells into your body to treat different diseases. And that one-time procedure should hopefully yield a cure for these patients suffering from these deadly diseases. It's really going to change how we think about medicine, but it's going to require an effort from the regulators, the health systems, the insurance companies, and the entire ecosystem to make sure that we can bring these transformative medicines to patients, not just in the developed world, but the entire world, so that we can change the lives of these patients.
Yeah. So it sounds like the promise of not just living longer, but also living more healthy. Sam, thank you so much for being with us today. It's just incredible the work that you and all of the employees and all of your researchers and physicians are doing. So thank you for your hard work, and thanks for being with us today.
Thank you, Ted.
I'm ready to take on the world. Ow!
In Ron Howard's movie Cocoon, senior citizens are rejuvenated by bathing in a pool storing alien beings in pods. Eventually, the aliens offer the elders much more than newfound vitality.
We'll never be sick, we won't get any older, and we won't ever die.
It's a metaphorical take on the fountain of youth, something humans have sought for millennia to no avail. Yet there's a growing number of people who seem to have found a version of that fountain, if not of youth, at least of longevity and relative health.
I can't really pick out which was my best days. They were all good, that's it. I'm Ike Newcomer. I was born 1911, and I'm 107 years old.
What's the secret to Ike Newcomer's long life? Certainly there's some genetics involved, but this profile of Ike and his family, produced by AARP, suggests something else that is also an important factor: family. 5 generations of Newcomers stay close to their centenarian patriarch.
I'm Dick. I'm 82 years old.
I'm Patty, and I'm 71.
I'm Dave, and I'm 53.
Chris, I'm 43.
My name's Devin, I'm 24 years old.
Tyler, I am 22 years old.
Jonathan, I'm 20 years old.
And then there's baby Knox, who'll be able to say he met his great-great-grandfather. These family connections aren't just good for Ike, they're a powerful model for what our later years can be like.
There's actually a future in getting old, and I think that's like a really optimistic way of looking at it that I've never thought about really until this very moment.
I don't really worry about getting older or what am I going to do tomorrow, what am I going to do next week. I just don't worry about it. I'll live for today.
Sadly, though, not all of us will be lucky enough to have such a caring and connected intergenerational family. So, how to create a version of that for the rest of us?
St. Anne Center for Intergenerational Care provides community-based daycare for people of all ages and disabilities. I hope it's like their home away from home. I hope it's even better.
Sister Edna Lonergan explained her vision for the St. Anne Center in this video from AARP. It started out more as a place for people with disabilities, but as her staff expanded to include many single moms without childcare, Sister Edna said, bring the kids to the center, with unexpected benefits for the adults and the children.
The adults just blossomed. They were just more alive when the children were there. So I thought to myself, why are we separating? We have excellent early childhood education. Children are learning their colors. They're learning to count. They also learn to be with people, comfortable with people who look different than they do. For our adults, I think it breaks the monotony sometimes in people's lives and the loneliness and the isolation that many older adults feel.
I was born with cerebral palsy from birth. It's a lifelong condition, so there's no cure. Just being here is awesome. It gives me something to look forward to. It's worth getting up for in the morning.
I'm 77 years old and I've just begun. I gotta keep showing people that it works, and I'm meant to help people have the confidence to start them. I wish that everyone could have what the clients have that come here. You look so pretty. Because we do take good care of them. They are loved, and they have a good time here.
This type of intergenerational program may or may not help Sister Edna's clients live longer, but it's certainly an example of the old saying, it's not the years in your life, but the life in your years that matter. Joining us for a conversation on healthy aging with dignity and purpose, please welcome Joanne Jenkins, Chief Executive Officer, AARP, and moderator Meredith Vieira, Executive Producer and Anchor, Founder and CEO of Meredith Vieira Productions.
I care so much about not just living a long life, because it's better than the alternative, obviously, but living a life that is healthy and a life of purpose. So when you talk about healthy longevity, what do you mean, Joanne? What comes to your mind?
Well, I think one of the things that we found here at AARP is that it's so important for people to live longer, healthier lives. And how can we as an organization who's out there supporting people 50 and older, how do we help them get to that stage that they are thriving in their 50 plus years? And that's about making personal choices in our own lives. I think we've all sort of stepped back during COVID to say, hey, I need to make some changes about how I eat, how I exercise. We know that people's personal choices about their own health, meaning eating healthier and exercising, actually controls 60% of their health outcomes, 20% based on the medical care they receive, and then another 20% based on their DNA, hereditary. And so we actually are in control of over 60% based on how we live our lives. And so for AARP, we're focused on providing safe and affordable healthcare, helping people get the content and information so they can be better informed. And as a result of COVID, much more attention on what it looks and feels like to be in a nursing home, having long-term care, as we've seen so many people have to deal with in this last year of COVID.
Yeah, so it's really interesting because on the one hand, COVID has been so isolating for all of us, and obviously many people have lost their lives or become ill. On the other hand, it has given us the opportunity to step back and really assess our health and what we need to do moving forward to live better lives and purposeful lives. So what are three things that we can do to help empower people to do just that?
Well, I always call it the health, wealth, and self, that we have to really focus on health security to make sure that not only people have access to health, but it's safe and affordable, that we have to look at financial resilience. The worst thing that could possibly happen is that we live this long, healthier life and not have the financial means to be able to financially afford that longer life. And then a self, that personal fulfillment. How do we address those issues of loneliness and isolation? We know that someone who is lonely, it's equivalent to smoking 15 cigarettes a day. The health outcomes really change the dynamics of your health outcomes if in fact you're a smoker. And so my shorthand for that is health, wealth, and self. How do we continue to help people focus on those three areas? And what can AARP do to make them more informed, more educated about the choices that they make in their own lives?
So let's break it down. Starting with health, what do you tell people?
Well, I think I'll give you a perfect example for myself, since I've been working at home this last year. And I used to tell people all the time, well, I would eat healthier if I wasn't on the road 3 days a week traveling and doing this. And so it was like, okay, I really have no excuses anymore. I really do need to eat healthier and exercise. And so I've started a walking regimen, eating healthier. And my health data is 100% better than it was when I was on the road traveling every day. And so that is sort of from that personal choice. In terms of the finances, so many people in this country are solely dependent on Social Security as their only means of income. And Social Security was never meant to be the only source of income. So how do we think about the fact that people 50 and over still want to work, Many of them are still in the workplace. They're still giving back, whether they have to work or whether they want to work. But how do you think about Social Security as part of a solution to having financial well-being? And then in that personal fulfillment space, how do we stay connected with friends and families? Obviously, we've all learned how to use technology much more efficiently with Zoom and Microsoft Teams and all these other different kind of software packages. But we used to think at AARP that, okay, we had a 3 to 5 year strategy about how do we bring people up to speed on the use of this technology. And COVID made us do that in 30 days, 60 days to really, bring the 50 plus population, many of who were already there, and using technology, not just to stay connected to family and friends, but to have medical appointments and do all of those things that we used to think you had to be there in person to do.
Yeah, I mean, I'm fortunate to have 3 adult children who I've called on so much during this pandemic, because I realized I really was not particularly tech savvy, and that's not a good thing in this day and age. You really need to get on the bandwagon and learn how to use it properly. But we're talking, and I love those choices that you've offered. But we live in a world that is not equal. There's a lot of inequity. How do you deal with that, the disparity?
Well, I think this past year has really brought to the forefront the health disparities and the financial disparities that exist, not just here in the US, but really around the world. And that we have to have that front and center and think about that in terms of how do we build a more equitable and inclusive society. I think about it in terms of what I call the 3 Ps, that we have to take a personal responsibility for showing up differently in our communities, in the workplace, in the way we interact with people who may or may not have the same benefits and upbringing that we have had, that we have to make sure that the private sector is stepping up to the plate, that corporations just can't sit back and continue to bring in all of this revenue from customers and yet not take a stand or put their money where their mouth is in communities to make sure that there are employment opportunities and really bring about a just society in our country.
How do you get them to do that?
I think that we've seen a lot of them stepping up because our employees are demanding it and society is demanding it. And we know that we see consumers making choices about what products they buy, what endorsements they make based on how those companies are actually showing up in the community. So this really looks and feels a little differently than some of these kind of incidences that have occurred in the past. So I think that's a good thing. And then the last piece is I call the public sector here, that the government at all levels, not just at the national, but at state and local governments, that we really do have to make adjustments and put in place regulations and guidelines that make sure that those who are underrepresented or the inequalities that exist in our communities, that we're addressing those issues that we can to make sure that everyone has a seat at the table and that we're hearing all of those different voices.
And if we don't close the gap, we really can't accomplish anything, can we, really long term?
I don't think so, because I think you will continue to see that gap, whether it's in the financial gap of the top 1% of earners in this country, and seeing the eroding of the middle class, particularly here in the U.S. And COVID, with so many people losing their jobs this past year, and so many in the service industry suffering as a result of closures due to the COVID. And here I'm at our headquarters at 601 East Street, and almost every restaurant in a 2-block area has closed down. And so, they're anxious for all of us to get back into the workplace so that they can revive their businesses. And, some will come back, but some won't. But I think that we all have a responsibility and can make choices to make life better for others around us.
Yeah, and I think so many people, Joanne, think of AARP and as well, that's for old folks, but you really take a multi-generational approach to aging, don't you?
Yes, well, and we know, particularly here at AARP and in many companies all across the world, that there are 4 to 5 generations in the workplace now. So think about this. A 10-year-old today has a 50% chance of living to be 104, assuming they have access to good quality health care and they eat well. That's amazing. That, you know, and that the first person to live to be 150 is likely already to be alive today. And that's because of increased knowledge in medicines and in the way we deliver our healthcare system. And so we're going to really have the opportunity to live, particularly our grandkids, to be over 100. The fastest growing age group in this country is people over the age of 80, and the second is people over the age of 100. And so we can't just assume, as our grandparents did, you know, years ago, that, okay, I'm going to retire at 62 and probably die at 68. People are living some 20 and 30 years longer. And we need to make sure that our healthcare system and our financial system, whether it's Social Security, Medicare, Medicaid, that those programs are revamped and revised to make sure that they're in step with our longer lifespans.
We have about 2 minutes left, a little under 2. And in that time, Joanne, can you tell us some of the things you're excited about that are happening in labs all over the country? I mean, I know ARP supports so many innovations and products that will make our lives better and more purposeful.
Well, we've been really aggressively looking at this through our AARP Innovation Labs, really trying to encourage startup companies to think about those areas of health and wealth. And some of the innovation that is coming through from these companies are around medication management. How do we remind people to take their medications, whether it's around home health care? How can we make it more convenient so that we can reach a loved one and be a part of those conversations through the use of technology with our parent or grandparents and in some of those areas where we need to see that additional assistance. It's also quite energizing because we see so many innovations or companies sprouting up because they're trying to address an issue that they're having in their own family, that they see it happening in their grandparents or in their parents. And we know one thing is for certain, you're either going to be a caregiver or need a caregiver. And so how we think about and how do we care for people who are older in society, I think is going to be key. And, you know, the excitement, not only in the healthcare area, but also in the financial area about how we deliver information and put in place protections so that people can continue to live in their own homes. We know that well over 85% of our members say they would rather age in place, that they'd rather stay at home than go into a nursing facility. And we know that the cost of that is much, much more cheaper. I won't say it's reasonable, but it's less than it would be if we were institutionalized in some kind of long-term care facility. So lots of excitement, lots of really good innovation coming from multiple generations to help solve some of these issues. So there's a lot of goodwill and a lot of positivity in our living our longer lives.
You know, I'm looking at you in that beautiful dress that says screams spring and hope, and you look so lovely and it brings a smile to my face. And that's, for me, that's the number one thing every day, just get a smile on that face and think positive thoughts and take it from there. Joanne Jenkins, what a pleasure to talk to you. Thank you so much for everything you do and AARP as well. And have a great life.
Thank you.
Yeah.
Richard Louie and Meredith Vieira join us to share how they balance family and career while providing nurturing care with dignity and devotion.
Hi, I'm Meredith Vieira, and I'm thrilled to be here with Richard Louie, MSNBC anchor, journalist, author, to discuss his caregiving journey. 53 million Americans, 53 million, are caregivers or partners in care for a loved one with a chronic disease. Richard, you're one of them, and I'm one of them as well. In your case, it's your dad, Stephen, and in my case, my husband, Richard. So I want to start out talking about your journey. It began, I guess, 7 years ago when your dad was diagnosed with Alzheimer's, your dad, Stephen. From what I understand, he had started to forget names of family members, which was not like him.
Right.
And that led you to have him see the right folks who were able to diagnose his illness. You made a decision. You were at the top of your game. I mean, this is in your dream job. And you made a decision to walk away from it or be willing to walk away from it to take care of your dad. I want to know how you reached that decision, because I would think it wouldn't be easy.
Meredith, this is great that we get to talk about this particular subject, because as many people as you have described are living through this, yet it's not part of our, as you know so well, it's not part of our day to day. In fact, we say caregiver, that's a strange word. Like that's just, I don't know what that is. In fact, we don't identify with it. And I had not done that in the beginning. I know that you at the same time had said, I don't know if that's really exactly my descriptor, my lower third, if you will, right now or all the time. And for me, I look to a lot of folks like yourself who did talk about it publicly in a very open platform, that you did have a person that would need you, does need you, and in different ways and at different times. And so for my father, when he could not remember his siblings' names, the ones that he had grown up with in a cottage, born in Chinatown, San Francisco, siblings, he had 12 of them. They're not even Catholic or Irish, but they still had so many kids, right?
Yeah.
And the story, poor, one fish, lots of rice, and a lot of gravy. That was them. And he forgot their names. And that was the beginning of his journey, and a very accepting and vulnerable journey that he shared with us with Alzheimer's. And there's no way that his kids were not going to lean in and try to help him along the way.
Yeah, in my case, Richard, when I met Richard,
Yeah.
he didn't, despite he told me he had MS on our second date, because he'd been with someone for a while, and when she found out, she sort of ditched him, and he didn't want to wait that long. He wanted to give me the out, because people were scared when they heard that. Most people didn't even really know what it was, but he didn't have what you think of as obvious symptoms of MS.
Right.
He didn't have any issues walking. He had some eyesight issues. And so I could walk into it thinking, well, this is going to be fine. And he had to explain to me, it's a long path. And I went with him to his doctor very soon after to begin to understand the journey that I would be on, maybe not in a week or a month or a year or two, and to begin to accept it. So I think, as you pointed out before, everybody's journey is a little bit different, and it kind of flows over the years.
Yeah.
Different ways.
And you said, I'm not a caregiver by that very descriptor. And what did you mean at that time when you were saying, I'm not a caregiver per se?
Because I felt more like a partner in care, Richard, and I still do, to be honest, although there are days when I definitely am the caregiver. He has bad days, and he has very good days when he's doing as much for me as I'm doing for him. And I also felt sometimes when you use the word caregiver, it diminishes the person who needs the care.
Yeah.
And I've seen that. When I see it, when we have travel, Richard uses a wheelchair, and people talk to the wheelchair. They don't see him. They see the wheelchair.
Yeah.
They say, well, does he want to do this? And I'll usually look at them and go, well, you can ask Richard. It's okay. So sometimes I feel that when I'm given the title caregiver, it allows people the opportunity to not see the person. I don't think it's done maliciously. I think people are uncomfortable with illness, essentially.
Yeah, and that's part of the gap, I think, right, yeah. Meredith, as you shared your story and I'm sharing mine about you're just not that one thing, both the caregiver and the caregiving recipient. And as I've, covered the story about caregivers in recent years, I realize I'm using sometimes even the wrong sentence structures. Maybe it should be a person that is caregiving. Maybe it's a person with a disability. In your husband's case, it's Richard. Richard is a person with MS, right?
Right.
That's structural to what you're saying when people see him in a wheelchair. That's a wheelchair person. No, it's a person using a wheelchair.
Exactly. It's just the way that you verbalize it. Exactly. But to go back to your story, what made you decide, I'm going to do this? I am going to be there for my father.
Like you, I guess as the journalistic nose that we've got, I did look into what's his journey going to be like. What did the neurologist say? I went with a couple appointments. I then before that though, I reached out to my boss, Yvette Miley, and I said, I think that there's going to be a time, because this is a long journey, 7 years on average, that he's going to be going through. And I don't want to miss a lot of those days. I want to be there for most of the days. And so when it came point when he was starting to forget how to get home or try to get out of the house or fall more, I knew I had to pull the trigger. They were very gracious to say, yes, we'll find something out of the box for you, Richard. And that was the warmth of our company.
Yeah, I think I was pretty comfortable with being obvious and honest about it. I mean, I stepped away because everybody has priorities, and my priorities were my family, which was more than Richard. It was our 3 kids as well, and making sure that that unit remained healthy and sound. And it was never, I don't care about my career, and I don't want to do anything but this. I wanted to make it work with my life, but my life revolved around my family. And you know when there are just so many hours in the day, as you say, even when we add an 8th day to the week, it's still, there are just so many hours. So you have to juggle. And that's what life's all about, whether you have someone with chronic illness or not. You're always determining your priorities, and they have to work for you. And I think once I settled in my own head, Richard, that this was right for me, I never lost a night's sleep. I was okay.
I find that I've learned how to cry better, and I've learned how to laugh better, and I've learned how to put it all together and realize that it is a 1 + 1 = 3, and that I would never wish it, just like you would never wish it upon Richard. I would never wish it upon my father, but it is required me to kind of re-identify who I am probably in the last 7 or 8 years 2 times. That was not on my wish list of things to do.
I didn't think of that, but you said your father really taught you what selflessness is. Taking care of him taught you that. What was that lesson?
Live and do what the right thing is, and don't beat your chest doing it. And yet, as you know, in our business, you kind of do have to bring attention to some of the things you do sometimes because you care about the topic, right? Like you and I care about caregiving. That's a very important topic. A family is something we both care about, and it's something we talk about a lot. So I think that's what I learned is, focus on the topic and sometimes bring in yourself, but not all the time. And that's really important. You have done in our space just about everything anybody wants to do. I know you just executive produced a great film as well, which is out. How's it affected your view on your creativity and your career?
I think it's made me, anytime you're following a path, it becomes very linear, and you're so scared to get off it. And I've gotten off it many times because of life's circumstances.
Yeah.
What I've discovered is it's the famous, the road less traveled. You find things about yourself that you never would have otherwise. I never would have done all the different things that I did with my job if life circumstances hadn't led me to be open to other opportunities. So I think in that way, it's been a blessing. What's your final take, Richard, from all you've learned in 7 years? When somebody says to you, what's a caregiver? What do you say?
Woo-wee. First of all, I get it. That is a tough word to understand. I will just say, it is the biggest job you ever take on for the longest amount of time, and will sit with you and give to you in ways you never thought were going to happen. And you won't be trained for it. You won't be paid for it in the ways that we think about it, right? But you will, for the rest of your life, remember that whatever you did was potentially imperfect, and probably imperfect, but in so many ways perfect for you specifically.
Well, Richard Louie, I really appreciate you talking to us. Thank you for sharing your journey as a caregiver and your dad's story. The book, Enough About Me, is out. When everybody views this, it will be available. So I hope people get it. It's really your journey and what you've learned about selflessness and lessons that we can all learn and should be learning during this period, actually. And also your documentary about young caregivers, Sky Blossom, is out as well. Richard Louie, thanks again.
Meredith, thank you. It was great hanging out and sharing our stories.
You too, kiddo. You take care.
Yeah, thanks.
We hope you enjoyed the first day of the 5th International Vatican Conference on Health, Innovation, Medicine, Science, and Faith. Today we explored where we are in the COVID-19 crisis and how we can build on lessons learned about preparedness, prevention, vaccine development and distribution, and how we can apply these lessons and principles for other chronic diseases. We looked at cutting-edge technology to repair damaged DNA and exciting new innovations to strengthen our immune system to help us fight disease. We discussed the mind, the body, and soul, and ways to reduce inflammation, improve dietary habits, and utilize the arts to improve our health and well-being. We learned about the impact that religion and faith can have on our health, and we dialogued about the need and ways to reduce the social disparities in health, how to help people age with dignity, and the upside of selflessness and caring for others who are seriously ill. We also celebrated two heroes, Stefan Bancel, the CEO of Moderna, and Helena Thurer, benefactor of the Hackensack Meridian Hackensack University Medical Center. They have influenced the lives of so many people through their ingenuity, leadership, and generosity. Tomorrow we will focus on the importance of dialogue and partnerships and explore the mind, the medical risks of loneliness, and the mental health epidemic. We will look at extending care into the community and into the home through technology and innovation, as well as bringing cutting-edge therapies to those who may otherwise not get access to such treatments. We will also examine new regulatory paradigms in regenerative medicine and advances in treating rare diseases, and will continue our discussions around aging and the principles of bridging science and faith. So please be sure to join us for day number 2. We will begin at 9 a.m. with a round table on human enhancement as part of our Bridging Science and Faith series. And we will follow this at 10 a.m. Eastern Time with opening remarks from Renato Poletti, the president of Science and Faith STOQ Foundation. Before concluding my remarks, I want to thank our moderators and our speakers for their participation. It is their incredible insights that make this dialogue so special. We also appreciate your help extending the dialogue by using your social media channels to share the highlights of the conference under the hashtags Unite to Prevent and Unite to Cure. Please remember to tag us at CuraFDN on Twitter, Instagram, and LinkedIn, and also on Facebook at the Cura Foundation. Lastly, we're so grateful to our collaborators, our grantors and supporters and benefactors, because without their generous support, we wouldn't be here today. They include Sanford Health, Cellularity, Akkad Holdings, the John Templeton Foundation, the Helmsley Charitable Trust, Sorrento Therapeutics, Aspire Capital, Hackensack Meridian Health, University of California, San Diego T. Denny Sanford Institute for Empathy and Compassion, United Therapeutics, Alliance Global Partners, Moderna, the Guthrie Jackson Foundation, AARP, the Eliza and Hugh Culverhouse Family Foundation, as well as Lawrence Krimker, John and Mary Papajohn, the Produce Marketing Association, Tivity Health, the Breton Family Fund, CRISPR Therapeutics, the Gary and Mary West Foundation, Healthy Villages, Horizon Blue Cross Blue Shield of New Jersey, JDRF, First Quality, Piper Sandler, Stern Aegis Ventures, The Church of Jesus Christ of Latter-day Saints, Cryoport, Dr. Randall Prost, the Susan Scott Foundation, Amicus Therapeutics, Arlene and Martin Bednar, Healthscape Advisors, Malik's Pizza Palace, Selective Benefits Group, and the Sovereign Order of Malta, and those donors who wish to stay anonymous. Have a great evening and get some rest, and we'll see you tomorrow.